AI clinical application in the biopharma industry session. My name is Tony, Director of the Clinical Diagnostic Scientific Affair at AstraZeneca. Wei Zhang, you want to introduce us?
Thank you. My name is Wei Zhang. I'm head of biometrics. I'm working for the Biopharma Therapeutics clinical stage biotech companies.
Thank you. So it's our privilege to serve as the moderators today for this exciting and timely discussion. So as we stand out the intersection of the bioscience and the AI, the digital innovation, the AI is rapidly transforming every aspect of the drug development. From discovery, translational medicine to clinical development and to safety monitoring and commercialization, right? Whether it's integration, digital and computational pathology, or the evidence AI is helping us to move faster and making smarter decisions. Today, we are honored to have three outstanding speakers with us and who will each share the insight from their work and the implementation in the different phases of the drug development life cycle. So, let's get started. Our first speaker is Dr. Yishan Chang. And Yishan is Senior Global Medical for Leaders Diagnostic at AstraZeneca, where he has the initiative in cancer diagnosis and pathology since March 2023. She joined AstraZeneca in the year 2021 as the U.S. Medical Director for Diagnostics, bringing deep expertise in oncology and precision medicine. And she graduated from Northwestern University with a PhD focus on system biology and macrophage biology. Dr. Shaw will discuss how to leverage computational pathology and use in clinical setting. With first zone, welcome Dr. John to the podium.
Thank you. Thank you so much, Tong, for the kind introduction. My name is Yishan Chang. As Tong said, I'm with AstraZeneca and I'm in the Medical Affairs Department. Today, my talk will mainly be focusing on the computational pathology to guide the use of ADC, antibody drug conjugates. These are my disclaimers and these are my disclosures. As an AstraZeneca employee, I do own stock. So I think it goes without saying that technologies have really changed the way that patients are being managed in the field of oncology. Many technologies are being applied currently in the clinic. To detect mutations, we have PCRs, we have DNA sequencing, RNA sequencing, and in many cases, immunohistochemistry, IHC, is being used in routine practice, such as identifying PD-L1, HER2 expression to guide patient treatment. In the field of IHC in particular, the current field is pushing towards the change and revolution into computational pathology. For computational pathology, it really all starts with the digitized image. Using digital pathology, scanning the whole slide image, we can look at the IHC scan slide down to the pixel level and extract the information and store this in computational processing power and analyze them using computational pathology. There are many facets of computational pathology. Chang went through this in his earlier talk in the clinical biomarker section, ranging between supervised AI, where we still rely on pathology annotation to individually annotate the slides, to weakly supervised AI, and ultimately to the foundational model. In the case of foundational model, or the self-supervised AI, essentially we are allowing the computers or the AI models to annotate them without the need of pathology supervision. You can imagine that this allows for a broader scalability in this type of biomarker development. If you look at digital and computational pathology, this is really not something that is only applied in the future. It is currently being used and being applied in the clinic in a wide range of patient journey. Diagnosis, screening, prognostication, accurate assessment of biomarkers, and ultimately to what we'll be focusing on more today, the new biomarker development. But before we go into the new biomarker development, I just want to go through some of the applications where AI tools are being used today. For example, MSI Intuit have developed improved pre-screening models for colorectal cancer using H&E prediction slides that you can have a sensitivity up to 92 to 95%, detecting the underlying MSI biomarker. This allows us to have 50% patients to essentially reduce the unnecessary reflex testing for MSI. In addition to that, we also see improvement in prognostication for patients with mesothelioma. And this is using the machine learning model to accurately predict OS for patients with mesothelioma. But how is this applied to future therapeutic targets? We have been talking a lot about antibody drug conjugates, ADCs. ADC is really reshaping cancer research today. ADCs, for ones for you that may not have heard a lot about this before, combines the precision of antibody targeting with a potent warhead. And in combination, this really allows us to drive precision in cancer research and have led to more than 15 ADCs now approved in the field of oncology. However, we're really not stopping there. One of the things that could be improved in the ADC development is a better biomarker development. This is where one of the AstraZeneca product, quantitative continuous scoring, QCS, could potentially come in to fill the gap. QCS is a computational pathology-based IHC image analysis that allows for an objective, precise, and measurements that's also reproducible. QCS at its core is really a two-step process. It starts with an image analysis where you precisely quantify IHC whole site image down to the pixel level. This allows the AI algorithm to first identify and distinguish between tumor versus non-tumor cells. And in addition to that, precisely quantify the protein expression on the subcellular component of individual slide and the whole site image. Using the richness of data that's extracted through the step one, for the image analysis, this information could then be applied directly and correlating it with the clinical outcome, which here you can see we can use QCS analysis to develop new biomarkers. One of the things, of course, with any AI developed models is this a black box? And truly, QCS is not. An example shown here as QCS, as I mentioned earlier, is a fully supervised deep learning model, which requires pathology supervision. Here you can see that pathologist annotation averaged 2.3 hours per field of view in order to identify the tumor versus non-tumor, as well as the quantifiable image. And this information is then fed into the model for training, validation, and testing. An example of how QCS is applied, you can see that there's multiple features that this type of information could generate for us. We can look at the percentage of positive cells. We can look at the density of positive cells. We can also specifically quantify the staining intensity of individual protein expression on each of the cellular components on membranes, on cytoplasm, or nucleus. And you can plot it on the histogram here in order to establish the cutoff directly correlating it with clinical outcomes. Using an example that I think we all are very familiar with, HER2. HER2 is a IHC testing that is already being routinely used in the breast cancer field for decades. And it's traditionally categorized in very discrete categories, IHC 1+, 2+, 3+. Here, what we're showing is that even within these distinct categories, when you apply digital and computational pathology, you really see the heterogeneity of these individual categories. Imagine that if these distribution does matter to patient response, we will have completely lost it without using a more sophisticated tool such as QCS. In addition to QCS measurements, we also have a very robust validation, including using mass spectrometry, as you know, a non-antibody-based protein expression measurement. As you can see here, what we measured with QCS very strongly correlates with what was measured and detected using mass spec, showing that this technology is extremely robust. One of the things I'm very excited to share with the audience today is one of our applications on TROP2. TROP2 is a very attractive therapeutic target that's been exploding in terms of its relevance lately. TROP2 is involved in many pathways within cancer progression. And in addition to that, it's highly expressed in cancer cells such as non-small cell lung cancer, and very low expression is detected in normal tissues, making it a very attractive therapeutic target. However, one of the challenges with TROP2 antibody drug conjugate development is that traditional IHC has not been predictive of response to TROP2 directed ADCs. And this could be due to various different reasons. And what we do know is that not all ADCs are created equal. Some antibody drug conjugates requires internalization for its drug cytotoxic payload to be released. Some simply release its payload as soon as it gets into the body. It's all about linker chemistry. This is where something like a technology like QCS can uniquely tailor the biomarker to the response. As an example here, and this was recently disclosed at the World Congress of Lung Cancer in Barcelona last year, we used QCS technology starting with the IHC-based TROP2 whole site image, scanned it, and looked at specific tumor TROP2 subcellular localization on the membrane and the cytoplasm. And what we did is used these values and created the biomarker called TROP2 NMR, normalized membrane ratio, which essentially is the ratio between membrane staining intensity to the total staining intensity combining membrane and cytoplasm. The biomarker is considered positive when a patient has more than 75% of the tumor cells with TROP2 NMR below 0.56. But this is not where we're stopping. Continuously, we're using computational pathology to approach many different development, including potentially multiplexing. And I think this is really a development that's dear to my heart, because specifically in lung cancer, more and more biomarkers are putting burden on the physician to use the very small tissue. Multiplexing really allows us to remove that burden and allowing patient samples to be used to the full extent. So with that, what I would like, if you have one thing to remember from my talk today, is really how exciting computational pathology and AI could be in transforming patient management. And thank you.
And to proceed, for us, and this is a certificate for you in also a gift. Thank you. Do you want to take a picture? Yeah, so we're going to take a picture at the booth there. Yeah, just
Like this?
all right. Thank you.
Yes, thank you, Dr. Chen, for that insightful presentations on the use of computational pathology to guide ADC development. A very powerful example of how AI is transforming decision making in precision medicine. So, because we are a little bit behind our schedule, so we will ask audience to keep their hold their questions, and we will have a panel discussion. So, we will the speaker will address the question as during the panel discussion. To attention to another critical applications of AIs in the biopharma space, pharmacovigilance. Our next speaker is Dr. Yuan Li, Senior Director of Biostatistics at Scholar Rock. Dr. Lin brings a unique combination of good statistical expertise and hands-on experience in developing AI-driven tools for clinical research. Prior to his current role, he was safety statistic at Moderna and whole research role at Merck. His work spans cultural influence, adaptive trial design, policy mindset, and integrated real-world and recognized data for regulatory decision-making. Dr. Lee is also active member of the ASA Safety Working Group and co-leads an initiative on combining randomized control trial and real evidence for safety evaluation. His talk today will explore how AI enhances both the efficiency and accuracy of observational research in pharmacovigilance and how regulatory guidance continues to evolve in this space. Please join me. Welcome, Dr. Lin.
Thank you, Dr. Zhang, for warm introductions. And also, this is my first time to attend the CBA. I was already really impressed by how organized this conference is being organized, and also how so many attendees. So this is really impressive. So today I would like to talk a little bit about leveraging AI and real world data in observation studies. So I really like to have an open discussion and also to see how we can learn with each other. So this is my disclaimer. And also I would like to acknowledge the contribution from the ASA Safety Working Group. So that we're working together to brainstorm ideas and also exchange how this is implemented across industries. So this is brief outline. It's really simple. So first I will briefly introduce the real data and real evidence, and then I will provide a landscape overview of regular guidance in real world data and also the AIs. And then I will provide several use cases, real use cases in the observing studies, and then we'll be concluding with final remarks. So for those of you are not really familiar with real world data, so real world data is just a data about the routine collected from various clinical practice, and the evidence is just generated from real world data. So there are multiple sources of real world data, including EHR, claim data, and registrations, PRO data, and more recently, the social media data, the health technology data will also be evolved. So comparing with the clinical studies, the study design is completely different, and also data is completely different, since we are using the secondary use of the real world data for research purpose, and the treatment is not randomized. It's based on physician choice and as also patient choice. And typical study design include case control, cohort studies, and they will also have the case reporting of case series. And still, RCT is the golden standard. and a different type of study design of these studies. They represent the different level of evidence, and the expert opinion case reporting is have provided the lowest level of evidence. So there are many area that real world data can be applied, such as the drug safety effectiveness, clinical practice guidance, market access, health economics. In particularly recent years, real world data have been really helpful in the regular decision makings, and it can provide supplementary information as with RCT, and when RCT is not feasible, so real world data can be support the regular decision makings, and they can answer a very broad stakeholder questions. For example, just pick ones. So for payer and insurance, so they are very interesting to how the treatment is cost effective and should at least be covered and in what tiers. And for pharmaceutical companies, also most evidence from the randomized clinical trials, but it's still we can have using real world data to better facilitate development the clinical development plan, and how in what's the landscape of disease, which treatment available, and what's the population we should target. So there's a range of questions that can be answered from with real world data. So next I will provide some landscape overviews. So for real world data and real world studies, so they already have a bunch of guidance from the academia and regulators. For example, STOBA guidance to help us to design a study, analysis, reporting, and how to interpret data. And especially we have recent years, and we have regular guidance from FDA, EMA about how to use that for the regular submissions. And also we have ICH guidance for the safety study, for the pharmacovigilance studies. And particularly in 2024, FDA have a final guidance on the assess EHR and claim data for supporting regular decision making. So in particular, this guidance acknowledged of using AI for the opposition studies. So it says that it can be used to better extract data element from unstructured EHRs, deploy the computer algorithms, evaluate imaging and laboratory data. So combining of AI and real world data, it's create a really powerful tools. And also in 2023 and revised in just this year. So FDA have discussion papers on using artificial AI in the development of drug and biological products. So in this guidance, it provides very nice overviews across different stages of clinical development, how AI and machine learning can be used, including the drug discovery, non-clinical research, clinical research, post-marketing safe surveillance, and manufacturing. So this guidance provides some insight about the consideration of AI in the regulatory in a regular settings. For example, the governance of transparency, how human-led governments can ensure the ethical use, and how we can address the bias, and also integrate also the privacy representativeness, and also it provides some discussion how we develop models, ensure the performance, and how we monitor the tools. And just early this year, there is draft guidance released by FDA. So particularly this guidance promote a risk-based credibility assessment approach for us to evaluate the tool and how it can be intended to support the regular decision makings. So this is a 7-step approach. First, define the questions and define the content of use, assess the AI models, and develop a plan, execution plan, and document the result, and also determine adequacy of AI models. And the main focus is all the regular decision making, but the model described can also apply to all non-regulatory settings, because we don't want AI to be misused, and if AI provide a bad result and it provide it's not able to replicate it and it's unethical, essentially it's no use for us. And EMA have a reflection paper, so it's very similar with FDA's. So it also outline potential use. So one difference is that it emphasizes more on the precision medicines and product information and also post authorizations. So it also provide a risk-based approach. So it categorizes, for example, the drug discovery as low regulatory impacts, and it grade all the activity in the clinical trial as high regulatory impacts. And also it discussed several technical aspects, and also it discussed the governance of AI. For example, GS, it needs to follow the GS principles, and how we could develop the standard procedures, and how we can ensure the compliance, and how we can do the human on-site and validations. And it needs to be a regular monitoring once it will deploy the tools. And in the pharmacovigilance areas, so there's CM's draft reports just released early last month. So it discussed the integration and implication of AI in the pharmacovigilance. And it provides several principles when we develop use AI in the pharmacovigilance areas, such as risk-based approach, human on-site, validity, robustness, transparency, data privacy, fairness, equities, governance, and accountabilities. So since when we're doing in the clinical development phase, the pharmacovigilance group, when we need to ensure the safety of patient, so it focuses a lot on these areas, comparing with the FDA and the EMA guidance. So now I will provide several use cases how this can be used in the observing studies. So first use cases is individual case safety report. So this use cases also be described in the FDA discussion papers. So you can see from this chart, there's a large volume of case report received from FDA and also sponsors. So which bring really huge burden for company to assess all those cases to make a quality of judgment and how we can submit to regulatory and essentially we need to protect the patient in the clinical trial, and after it's put into a market, we need also ensure the product is safe to use. So this is the flow chart. So first it take large, different kinds of data from spontaneous reporting, clinical trial, EHR data, social media, literature, and registrations. All those different data are in the different structures, and some field they may have missing data, And so AI can detect the patterns in all those data set, and it can put it into a more structured ways that we can analyze. And also it can perform validity check before we proceed to those cases, and also duplicate, and then we can put in a coding, and then we can in-feed to the next stage for the case evaluations. So AI can also help us to perform the clinical assessment based on the cases we received, and also based on the literature that they can gather together. And then it can perform the causality assessment based on the causality framework, and to make judgment whether this is related or is unrelated. And also it can make an outcome assessment to see whether it's a serious event or a non-serious, is expected or unexpected, so that it put it into the right category for the FDA submissions. And together with aggregate safe reportings, and we could it could be compile the report for the submissions. So the next use cases is about the unstructured healthcare data. So by estimation, there are around 80% of healthcare data is unstructured. It's hidden in the clinical notes, imaging report, or physician narrative. So it's difficult to analyze if it's unstructured. So how we can put it into the structured data? So it involves a lot of physician reviews and also educations. So with AI, it can use the larger language model to pre-process the data to put them together, and also put the add annotations and establish a model, and then we can proceed a lot. And also human oversight can be put in together. So they will also need human oversight. So we can, if there's any discrepancies, there will be educations. In addition, so the real data in different countries, they are from different clinical practice, and the coding and also the healthcare system is also different. So the AI can help us to identify those patterns. So put all the different countries' data into the same common data models. So this is just one example. So this is a patient have this is a physician note. So it have basic information about this patient experience and also the medication use. So AI can detect. I mean, we provide information about the structures we need to extract, and then we can put all those related information into a data set so that it's ready for us to do the analysis. Next use cases is about literature review and evidence synthesis. So right now they have a variety of AI tools developed for the systematic review and evidence synthesis. So if you search, you do a search there, and you will find a lot of tools. So it can help us to do a lot of things in different phases, such as develop a search strategy, locate relevant articles, and also during the processing that can help us to screen the data, extract data, and then synthesize them together. So there are two guidance. You can take a reference. So here is our flow chart. So first, the reviewer can define the scope for this research, and define the keywords and what's the database you need to research. Then AI can take that to extract, to set up the queries, and then it gather all the reference and read the abstracts, and find the PCOS criteria such as the population, intervention, comparator, outcome, and study designs. Then it can take a lot for the abstract screenings, and it can have assess the relevance and exclusion criteria, and remove duplications, and also can help us and then we can compare result with human result, and then we can make educations, and after that we can get all the information is fill into the criteria, and then we could do the more detailed data extractions, and then we can do the analyze, summarize them together. And all this can be done automatically once we set up the protocols, and it can automatically extract new information, and then we can have a more updated result. And next application is about the study design analysis. So the AI tool can help us develop the study protocols. So we will only need to provide the training data set from using the historical data, and then ask the AI to generate a protocol based on the study synopsis and provide a standard template. So this is still in the active research. I mean, because we still need to make it more useful. For example, for the technical difficulties, AI still have some difficulty to understand the complicated study designs, and also how we can interactive with AIs and doing version controls, and how we can do the risk mitigation to make sure it's not biased and biased out to a particular study population, etc., etc. So there's still some work to do. And in the observation studies, so particularly it will be really useful for the target trial emulations. So when we compare with RCT and observation studies, over time we said it's apple to orange comparisons. So it's difficult to explain if there's any discrepancies. So target trial emulation is a method that can emulate the trials, such as the study objective, the estimate, and the analysis method, so that we could make the study result among comparables. So AI tool can better understand the pattern hidden in observation studies, observation data, and so that we can help us to better emulate the trials. For example, AI can access the free text so that it can identify the eligible subject not considered initially by the humans, and also it can also help us exclude certain group that may not be included in the RCTs. So this is another example that we could use AI to help us to develop a statistical analysis plan. So similar to the study protocols, we input the historical data and ask to provide based on the SAP template. It can generate from the outline to the full SAP. And also it can generate the statistical programming for us to execute. Yeah. So that's the final remarks, and so I think I'm on the right time. So in this talk, I briefly discuss the regular guidance and how it can be help us to shape the AI tools, no matter it's in the regular environment or not. And also I provide several use cases for the observation studies. And AI tools, so it can bring down the cost and also speed timings when we need to, in a traditional way, to design observing studies, and with appropriate human oversight and validation of tools. So it can generate continuous evidence, so that in different setting, in some settings it's really attractive. A lot of times, sometimes we need to quick answers. We need to develop. We need speed clinical development. We need information to support decision makings. So instead of set up observation study, take a few months, AI tool can speed this process. And another one is I want to provide my personal view of how AI tool developed it in healthcare. So currently we have AI tool of large language models, and recently retrieval augment generation is a tool can augment the answer with local data so that it's more tailored to our needs. So and in additionally, the model context protocol is involved. Just came out 4 months ago. It quickly go follows, and every IT company is talking about this. So this method can help us organize large amount of local data and also connect different AI tools together. So this will be the traditional method that in the future there will be a personalized AIs, and also automation make it possible. So we can foresee that in the future, this can help us to develop the AI automation in healthcare for certain areas. But in the healthcare, we know this is a heavily regulated environment. How we can integrate it into our process, how we can interact with humans. So integrated AI and the human health system, may take more time. So that concludes my talk.
Thank you, Leon. On behalf of the executive committee, we would like to present this certificate to recognize your contribution to the CBA annual conference. Thank you.